Hello everyone, welcome to our new section. Today we are going to discuss about the classification and structural hierarchy of proteins. Proteins are the nitrogenous organic compound of high molecular weight which play a vital role in the living organisms. These are made up of 20 amino acids. They serve as a bodybuilding units and also provide support and protection to various tissues. Proteins also help to regulate the physiological or cellular activities. Large scale study of proteins are known as proteomics. Proteins are classified based on their source, nutritional value, shape, solubility, function, and composition. Then, classification of protein based on their source. Proteins are classified into animal proteins and plant proteins based on their source. First, animal protein. Proteins which are derived from animal sources such as egg, meat, fish, and dairy products are called animal proteins. They are also known as higher quality proteins because they contain sufficient amount of essential amino acids. Some nutrients are more abundant in animal protein sources such as vitamin B12, vitamin D, DHA and zinc. Next is plant proteins. Proteins that are derived from plant sources such as cereals and nuts are known as plant proteins. They are also known as lower quality proteins because they contain one or more essential amino acids. Plant contain common amino acids are lysine, methionine, threonine and tryptophan. Next classification is based on their nutritional value. Proteins are classified into complete, incomplete and poor proteins based on their nutritional value. Complete proteins. These are known as nutritionally rich protein or first class protein because they contain all the essential amino acid in required proportion. Casein of milk is the example of complete proteins. Incomplete proteins. These are the proteins which lack one essential amino acid. They cannot promote body growth in children, but they can maintain the body weight in adult. Protein from pulses are deficient in methionine and also protein from cereals lack in lysine are the examples. If both of them are combined in the diet, adequate growth may be obtained. Next is poor proteins. These are the proteins which lack many essential amino acids. Diet based on these proteins will not maintain original body weight. Zein from corn, lack, tryptophan and lysine are the example of poor protein. Next classification is based on their shape and solubility. Proteins are classified into globular and fibrous protein based on their shape and solubility. First one is globular protein. These are the protein which are spherical or oval in shape. In case of solubility, they are easily soluble in water. Blood protein like serum, hemoglobin, antibodies are the example of globular proteins. Second one is fibrous protein. These are the protein which are elongated or needle shaped. Their solubility power is minimum. They have both structural and protective roles. They resist digestion. Proteins of connective tissue, bonds and blood vessels are the example of fibrous proteins. Fibrous protein again classified into collagen, elastin, keratin and fibrin. Collagen. Collagen are the proteins which is rich in hydroxyl protein. More than half of the total protein in mammalian body is collagen. Keratin. These are the main constituent of epithelial tissues. They contain large amount of sulfur in the form of cysteine. Human hair contain about 40% of cysteine. Elastin. Elastin are the protein which are forming the main constituent of connective tissues and especially found in the dermis of skin. They provide elasticity of tissue. Fibrin. These are the insoluble proteins. They are formed from the fibrinogen during the blood clotting. These are the proteins which mainly composed of glycine, alanine and serine units. Next classification of protein is based on their function. First one is transport proteins. These are the proteins which is found especially in animals, which is involved in the transport of many essential biological factors to various part of organisms. For example, hemoglobin carries carbon dioxide from tissues to lung and oxygen from lung to tissue. Ceruloplasmin transport copper in the blood. The blood plasma contain lipoprotein which carry lipid from the liver to other organs. Another examples are myoglobin, albumin, transferrin. Next is storage proteins. These are the biological reservoir of metal ions and amino acids. Albumin is the major protein of egg white. The milk protein casein stores amino acids. Third one is regulatory proteins. These are the proteins which may regulate the cellular or physiological activity. Example are insulin and growth hormones. Fourth one is catalytic proteins. These are the enzymes which increases the rate of reaction in our body. Some of them are simple or complex. Urease, amylase, catalase are the example of catalytic proteins. Fifth one is genetic proteins. Genes that contain the information needed to make functional molecules are called genetic proteins. Histones are the example of genetic proteins. Sixth one is toxic protein. The protein which are toxic to our body known as toxic proteins. Bacterial proteins and snake venom are the example of toxic proteins. Seventh one is structural proteins. These are the proteins which play an important role in shaping the skeleton and structure of cell. Collagen, elastin, keratin are the example of structural proteins. Eighth one is contractile proteins. Some proteins which are help the organ or cell to contact change in the shape for the easy movement. Actin and myosin are the major contractile proteins in the vascular smooth muscles. Last one is protective proteins. These are the proteins which is produced by immune system to respond the presence of foreign substances. Fibrinogen and thrombin that prevent the loss of blood are example. Another example are immunoglobulin and interferon. Last classification of protein is based on their composition. Proteins are classified into simple, conjugated and derived proteins based on their composition. First one is simple proteins. These are the proteins which is made up of only amino acids which are joined together by the peptide bond. There are 7 types of simple proteins. They are albumin, protamine, prolamine, globulin, lectin, glutenin and scleroprotein. Second one is conjugated protein. Besides the amino acid, these are proteins that contain a non-protein moiety known as prosthetic group or conjugated group. The prosthetic group may be metal or a compound. They are attached by either covalent bond or other interactions. There are 6 types of conjugated proteins. They are lipoprotein, nucleoprotein, phosphoprotein, chromoprotein, metalloprotein and glycoprotein. Third one is derived proteins. These are the denatured or degraded products of simple and conjugated proteins as a result of partial hydrolysis by the action of acids, enzymes or alkalis. These are classified into primary and secondary proteins. Primary derived proteins are 3 types: proteins, metallo or infoprotein and coagulated proteins. Secondary proteins are 3 types: proteoses, peptones and peptides. Next, we have to discuss about the structural hierarchy of proteins. Proteins are structurally organized into 4 levels: primary structure, secondary structure, tertiary structure, and quaternary structure. Primary structure of protein. Primary structure of protein refers to the sequence and arrangement of amino acids in the polypeptide chain. The carboxyl group of one amino acid is linked with the amino group of another amino acid by the peptide bond. In polypeptide chain, the carboxyl group and amino group of most amino acids are involved in the formation of peptide bond. However, Two amino acids which are situated at either end of polypeptide chain have either carboxyl group free or amino group free. The end at which the carboxyl group is free, known as C-terminal, and the end which is amino group is free, called N-terminal of the polypeptide chain. The peptide bond occur in trans configuration and have a partial double bond character. Due to the partial double bond character between the carboxyl and amino group, they do not rotate during the formation of folded secondary and tertiary structure. Second one is secondary structure. The conformation of polypeptide chain by twisting or folding is referred to as secondary structure. The amino acids are located close to each other in their sequence. There are two types of secondary structure, alpha helix and beta sheet. Alpha helix. Alpha helix is the most common spiral structure of protein. It has a rigid arrangement of polypeptide chain. The alpha helical structure was proposed by Pauling and Corey in 1951. The alpha helix is a tightly packed coiled structure with amino acid side chain extending outward from the central axis. These are stabilized by extensive hydrogen bonding. The hydrogen bonds are individually big, but collectively they are strong enough to stabilize the helix. All the peptide bond except the first and last in the polypeptide chain participate in the hydrogen bonding. Each turn of alpha helix contains 3.6 amino acids and travel a distance of 0.54 nanometer. The spacing of each amino acid is 0.15 nanometer. Alpha helix is a stable conformation formed spontaneously with the lowest energy. The right-handed alpha helix is more stable than the left-handed helix. Certain amino acids disrupt the alpha helix. Large number of acidic or basic amino acids are also interfere with alpha helix structure. Beta pleated structure. This is the second type of structure proposed by Pauling and Corey. Beta pleated sheets are composed of two or more segments of fully extended polypeptide chains. In the beta sheet, the hydrogen bonds are formed between the neighboring segments of polypeptide chains. The polypeptide chains in the beta sheet may be arranged either in parallel or anti-parallel. Third one is tertiary structure. The three-dimensional arrangement of protein known as tertiary structure. It is a compact structure with a hydrophobic side chain held interior while the hydrophilic groups are on the surface of protein molecule. This type of arrangement ensures the stability of molecule. Hydrogen bond, disulfide bond, electrostatic bond, van der Waals bond and hydrophobic bonds are responsible for this protein structure. Last one is quaternary structure. It exists in protein consisting of two or more identical or different polypeptide chains. These are known as subunits. These proteins are also called oligomers because they have two or more subunits. The quaternary structure describes the manner in which subunits are arranged in the native protein. Hemoglobin and lactate dehydrogenase are the example. Thank you.